Thank you. All right, so for bonus, module down at the bottom of our modules page. So again, thank you so very much to those of you guys last minute that came and helped out for Delgado Day. Make sure that you click this link, you know, let me know your time, and I'll give you your points. This bonus activity right here, I'm going to go ahead and change the settings in that to that you guys can upload a file, which means that you can take a picture of the notes that you did, and then when you click submit, there should be the option of uploading a file. In other words, uploading your pictures. Does that make sense? Okay. So that way, because again, for this assignment, I want you to take notes. So if you're not going to type them on the computer, if you handwrite them, go ahead and just take a picture of them and then upload that picture to get your points. So I asked you guys to watch those before class today, and if you did, this is going to help you, right? I don't do these things to punish you guys, right? I do these things to help you, because one of those was a really good review, right, of what we covered last time, these last two on antibody action, right? Should have hopefully been a good review for you guys. So if nothing else caused you to review your material. This other stuff was new stuff, right, which I'm going to be going over today. So it was a good almost introduction to today's lecture. So you still have time. You'll notice the deadline, I'll just go back to the main page, isn't until I think next Thursday or Tuesday, right? Probably Tuesday. Yeah, Tuesday. So, you know, now that I changed that to those of you guys that already did it, you can just take pictures of your notes and upload them. Those of you guys that haven't done it yet, you still can do it, right? And I definitely encourage you to do so. Those are some pretty good videos, I think, anyways. So I posted one of the other bonus opportunities for you guys, which is next Monday, right? And it's another one where, again, take notes. So you know what? If you want to just take hand notes and submit those, I'll let you guys do that too. Upload a file. Done. All right? Cool beans? All right. So let's get to the cool stuff. This is some of my favorite stuff that we're going to talk about today. So. Huh? Oh, yeah, but we have other stuff, right? These aren't the only things. I know it sucks when you have something else you gotta. It was like we were supposed to have a seminar this past Monday, and it got canceled. And I was like, oh, good, I can do this then. And then my boss followed up with an email like, lead instructors have to meet still. And I was like, really? Really? I was really hoping to get some work done. I was even thinking about skipping the seminar, but, you know, it was like, I wanted to go. It's hard. It's hard, right? Juggling things, and you can't be at two places at the same time, unfortunately. So, but we do the best we can. So we've been talking about the adaptive immune response. And so adaptive, remember, think about change. You're born with this, just like you're born with all your components of your innate immune. And as we'll see, a lot of the components of your innate immune system, as we've already seen, interact with adaptive, right? So these aren't separate systems, but they do have some differences. And one of the major differences with adaptive is its specificity, that level of specificity that we don't see with innate. So it's because of its specificity, is probably the most effective at eliminating invaders. The problem with it is that it takes a week or more to build following the first exposure. So the very first time you've ever seen this antigen, it's going to take your body about a week to go to this level of response. So in that time frame, your innate is got to keep you alive, basically. The sad thing is that sometimes your innate is not enough, right? and you don't survive this attack by a microbe. But one of the cool things too about the adaptive is its ability to remember, to have memory. And we've exploited this with vaccination. So what vaccination is maybe your first exposure, but in a way that you should not get the illness, right? So we've given you a weakened form of the virus, or we've given you a weakened form of the bacteria. It may even be killed. It may even just be pieces of the bacteria, right? We may give you the toxin that something produces, right? But in a way that it's not toxic to you, but your immune system gets a picture of it, right? Sees what this molecule looks like. So that way, the second time you're exposed, one of the cool things about this is that is much stronger. And also, you're going to not have to wait a week, right? You already remember you've seen it before. So you don't have to worry about the potential of the innate failing or not being able to take care of the problem. So in the case of tetanus, right, that causes that bacteria, which is an anaerobe, when it gets into a wound that's gone anaerobic, and say those spores from the environment got into that wound and it germinates and it starts growing. Well, unfortunately, it produces a toxin that can then get into your bloodstream and spread throughout your body. That toxin stops your inhibitory neurons from telling your muscles to relax. So instead, they stay contracted. That toxin is what you get vaccinated against, right? So they give you a weakened form of that toxin, and what do you make against that toxin? You make antibodies against that toxin, right? Antibodies are the most common product of your adaptive immune response. And the thing is the cells that made these antibodies remember that they made it. So if they see it again, or when you get boosted, your levels go up very quickly. The response is much quicker the second time because you have cells already that recognize. And you may even still have antibodies around that recognize that specific foreign substance, right? That tetanus toxin. And as we saw, as we looked at antibodies, right, that interaction is at the molecular level, right? And we're not talking about an antibody binding even to an entire protein, right? Just little sections of a protein, like 10 amino acids. So in fact, you don't even make just one antibody against the tetanus toxin. You could make several different antibodies. Right? We're not going to just shoot them with one bullet. We're going to just obliterate them, right, as best we can. And then last time, right, and I'm not going to review again, but again, that bonus videos to go over it again, all the protective outcomes that are potential with the antibodies, right? And then we talked about last time all the different types of antibodies we make, right, that have different jobs based on where they are in the body, or in the case of IgA, is it even in our body? No, technically it's outside, right? It's secreted. It's in our mucus and our breast milk and our saliva and tears. The other important thing about this system is being able to recognize healthy self as opposed to something that's dangerous. Because this, and the reason why we say healthy self is because we can even recognize when there's something wrong with our own cells. Right, with the adaptive immune response. And typically that's done by T cells, where B cells are the ones that make antibodies. And so we're going to start talking about these guys. And so even within adaptive, we kind of have a division. We talk about humoral immunity versus cellular immunity. So humoral immunity is antibody immunity, basically, right? Some people just refer to it as antibody immunity. when scientists were first studying the immune response, one of the easiest things for them to detect and see were the antibodies, right? They're proteins. They get secreted into our secretions, as we just said. They're also all throughout our blood. And so, especially when, when they studied blood and things like that, they found these proteins that had this protective outcome. And so this was referred to as the human element, right? The humoral element, until we assign the name of antibodies, or sometimes immunoglobulins. Cellular immunity is cells, right? Detecting problems with cells. And this is important, right? Because where do viruses get? Antibodies are only going to help us when the virus is outside the cell, right? But where does a virus need to go? Inside of our cells. Well, once that virus gets inside, what protection do we have from innate immunity that's going to help us maybe detect a virus got in? What's going to help us from our innate immune response? What do we learn about as it relates to viruses? What protection do we have against viruses? There's receptors, right? There's receptors inside of our cells, right? And they're going to release a particular type of cytokine to inform other cells that there are viruses present. What's the name of those cytokines? It's a particular class of cytokines, specifically for viral infection. Interferon. Interferon. Put that one in your memory bank. Right? Interferon. So we have multiple levels of protection, right? We have interferon that's going to help, but two, if a virus gets inside, a lot of the viruses have got to put their proteins into our membranes to make their envelope out of our membranes. When they do that, if we have antibodies that recognize those proteins, and the antibodies can bind. But remember, then the antibodies themselves, do they directly kill a cell? No. What cell is going to be needed to help kill that cell? Who kills cells that are coated in antibodies? Especially if it's IgG antibodies. ADCC. It's one of your antibody-antigen interactions. Natural killer cells. Well, you didn't say it loud enough. You guys screaming at me. Be loud and proud. I'm still stuffy. My ears are stuffed up too. Okay. So, that's good for you, though. I didn't do my little quiz thing for today, as you guys can notice. Otherwise, I'd have you typing it in. Okay. But T cells, too, have receptors on them, where natural killer cells are really dependent on that antibody doing that job. But T cells have receptors that can specifically recognize pathogens associated with cells, right? And so we have different types of T cells because of that have different jobs. In this picture that I stole from the first website that I sent you guys, right, the really cool thing, like I said, about this system is they specifically recognize an invader, right? Right down to the molecular level. So this guy is probably a B cell. These are probably antibodies, although that's not how I would draw an antibody, right? Because they're only really showing one antigen binding site, where antibodies, remember, are that Y shape, and they have two antigen binding sites. But the reason why I say that this is probably a B cell is because this is an antigen, right? I think they say in the thing it's like a bacteria. And again, bacteria are going to have different molecular components associated with them. So these cute little crowns they got on this guy, see how each one of them is shaped a little bit differently? That's to denote a different epitope on them, right? A different antigenic determinant. And antibodies only bind to specific things, right, on antigens, right, to those antigenic determinants. And one B cell, all its antibodies are the same, right? All its antibodies are the same. So he can only recognize one specific pathogen. So this one cute little B cell only recognizes this one bad guy. So he says, you know, this one fits. But do you only have one B cell in your body? No, you make millions of them, right? And each one of those B cells is different. Each one of them is different. So each one of them can potentially recognize their bad guy. Does that make sense to you guys? So the reason why I know this is a B cell is because only B cells interact directly with an antigen. T cells, on the other hand, interact with other cells. This isn't a body cell, right? This isn't an immune cell. This is a pathogen all by itself. Ugly little bacteria. So we're going to watch a short video. And there's one mistake in here that I have to admit I didn't notice for the longest time. And I really should write the publisher, but I don't know if they could fix it. But I'm going to let you guys know it. Okay? I should give it as a bonus one of these times. Be like, what's wrong in this animation, right? Really test you guys. All right. It's minor. It's a minor thing, but important even still. Oops.
Typically begins when a pathogen enters the body.
Let's start over.
Activation of the immune response typically begins when a pathogen enters the body. Macrophages that encounter the pathogen ingest, process, and display the antigen fragments on their cell surfaces. Macrophages with antigen fragments displayed on their surfaces are called antigen-presenting cells. An antigen-presenting macrophage interacts with a T helper cell that can recognize the same antigen. During the interaction, the macrophage releases a chemical alarm signal called interleukin-1, which stimulates the T helper cell to secrete interleukin-2. Interleukin-2 causes the proliferation of certain cytotoxic T cells and B cells. The immune response from this point follows two paths, one using cytotoxic T cells and one using B cells. Normal cells of the body that become infected can also digest some of the pathogens and display antigen fragments on their cell surfaces. The body makes millions of different types of cytotoxic T cells. Each type is able to recognize a particular antigen. The cytotoxic T cells that are capable of recognizing the antigen displayed on the surfaces of infected cells bind to the infected cells and produce chemicals that kill the infected cell. Death of the infected cells results in destruction of the pathogen. B cells also come in millions of different types, each able to recognize a particular antigen. When B cells become activated by T helper cells, they differentiate into plasma cells. These plasma cells become antibody-producing factories, flooding the bloodstream with antibodies that can bind to the antigen involved in this infection. Antibodies bind to the antigens on the surfaces of the pathogens, marking them for destruction by macrophages. Some of the B cells do not turn into antibody factories, but instead become memory B cells that may survive for several decades. Because of these memory B cells, the secondary immune response to a future infection by the same pathogen is swifter and stronger. This powerful secondary immune response is what gives immunity to some diseases after you have had them once or after you have been vaccinated.
It's pretty cool, huh? Until we start getting to the specifics, and then you guys are like, ah! All right, so let's rewind. So who is this? What cell was this? No, very beginning. Macrophage, right? And who else does this, and actually does this probably more so than a macrophage, ingest material, and then the next step is he did what? He presented it on his surface, right? So there he is presenting it on his surface. Anyone know another cell that does this? Dendritic cells, right? Those are your main guys usually for antigen presentation. But macrophages can do it too. If you guys want, you can close the door. It's noisy. It's always noisy. Thank you. So, now I'm going to really quiz you guys, especially you guys that watched my videos. Anyone know what the name of this molecule is that does the antigen presenting? It's an abbreviation. MHC2, which stands for Major Histocompatibility Complex 2. Guess why it's called this? It's called this because these receptors on the surface of our cells, right, our immune system uses to communicate with each other. When you go to give somebody else someone else's organ, those are different, right? Your how to make this receptor is encoded in your DNA, right? Just like everything your cells do is encoded in your DNA. So that's encoded in your DNA, how to make these receptors. And the thing is that you have cells that if they can't interact with these receptors, right, if our immune system can't recognize these receptors, they think the cell is bad, is foreign, and needs to be destroyed, which is true. If you got someone else's organ, it is foreign. And remember, part of this system is being able to recognize self, healthy self, versus non-healthy self, and definitely anything that's foreign. So that's why when they go to try and do, you know, organ transplants, liver transplants, kidney, heart, whatever it is, they want to try and match markers that are found on our cells. They're talking about ones like this one, right? And more so class 1, because class 1 is found on all your cells, right? So those liver cells you got from someone else are expressing MHC class 1 that's encoded by the DNA in those cells, which are not your cells. And so that's where it creates a problem, is the immune system goes, oh, that's not me, right? And goes to attack it. That's why they want to try, I don't know about you guys, but I don't have an identical twin. I mean, I've got peeps out there that probably look a lot like me, right? But I don't have an identical twin. So there's no one out there that's genetically identical to me, right? I don't have a spare parts person, right? They have those weird sci-fi movies where they make cloned people for spare parts. You know what I'm talking about? No sci-fi peeps in the class. Okay. Oh, there's, you know, I mean, we probably will be there someday where they'll make a whole nother human being clone for you to have spare parts. But we're not at that point, and probably they'll probably ethically stop us from being able to do that. But so we're going to try and match as close to you as we can. And so, of course, close relatives like your parents or your siblings are most likely to be the closest match, right? Because genetically you're similar, right? But you're not going to be the same. But then if you don't have that, then, you know, that's why, you know, matching, right? Getting these matches can be very difficult for some people. Because you want to try and match it as close as you can. So unless you've got a perfect match, you've got an identical twin and they give you their extra kidney or something, you have the potential of your immune system attacking and destroying that organ that you need to live. So guess what the people who get organ transplants have to do for the rest of their lives? Take medication, but what does that medication do? It suppresses their immune system so it won't kill the organ. Because you wouldn't get that organ if you didn't need it to live, right? So it's a matter of living, but you're sacrificing what? Your immune system, right? People who get organ transplants, trust me, they don't go work in daycare centers because those little things are germ magnets, right? Your immune system is compromised. They don't go to festivals with thousands of people, right? Because, you know, the risk of infection is so much greater under those circumstances, right? They have to be very cautious because they're purposely weakening their immune system to keep the organ that's not theirs. Does that make sense to you guys? Okay. So these markers are very important for this communication, right? This specific communication with our body. So MHC class 2 is important for antigen presentation. So antigen presenting cells have it. So we've already said this macrophage has it, right? Dendritic cells have it, and then there's one more that we see later in this presentation. Anyone know? Does antigen presentation. The B cell. The B cell interacts with the antigen via their antibodies, right? Then they're going to internalize it, and they're going to present it on MHC class 2. Okay, now the next important step. Who does this cell talk to? The T cell. More specific. T helper cell. So here's that interaction, right? That's a helper T cell. What does the T cell have on its surface? A receptor, right? This one's easy, y'all. It's the T cell receptor, right? But again, this receptor it has is specific to that antigen that this macrophage is presenting. This is a specific interaction. And again, you make bunches of different T cells that all interact with different antigens. And how to make that T cell receptor is encoded in their DNA. What they don't show you here is there's another receptor here that allows helper T cells to talk to antigen-presenting cells, ones that are presenting MHC class 2. So I almost think of it as like a secret handshake, right? Because MHC class 2 and helper T cells, that's it. That's the secret handshake. Those are the only two that talk to each other. So there's another marker that we found on immune cells, and there's lots of them. So they're abbreviated CD, which actually stands for clusters of differentiation. So again, this was this whole grouping of receptors on the surface of our immune cells that allow the different immune cells to do different things, and also allow for us to see this difference between a helper T cell versus, say, a cytotoxic T cell that we mentioned later. So helper T cells, a lot of times by scientists, are actually referred to by that marker. Anyone know what that marker is? What number, what CD is a helper T cell? Helper T cells are CD4. This is the cell that gets attacked by the HIV virus, y'all. And this is why a lot of times, right, people who have HIV infection, they'll run special diagnostic tests to see how many helper T cells that patient has. And when those numbers get very low, then you progress to the status of what we refer to as AIDS, which stands for acquired immunodeficiency syndrome, because at one time we didn't know what caused it, right? And other things can cause it, and you can truly say, although AIDS has been specifically linked to HIV infection, people who have organ transplants basically are in an AIDS state because they have an acquired immune system that has been deficient because of the drugs. But in truth, they don't give them so much drugs that they really reach that status of what we say as being deficient, but definitely compromised. Right? But that's where that terminology AIDS comes from. It means acquired immunodeficiency syndrome. So there are people, because of drug treatment nowadays, living with HIV infection, right, and haven't progressed to that very low status of their immune system being compromised. And that isn't actually what kills them, right? It's the fact that their immune system can't protect them, so they get some other infection that their immune system can't take care of, and that infection actually is what kills them, ultimately. What's the most common infection that AIDS patients get and kills them? Pneumonia. Number one killer of AIDS patients, pneumonia. And think about it, it's something you inhaled. How do you really avoid that? So, yeah, we can specifically test to see how many CD4, how many helper T cells do you have, right? And obviously you want to have a lot of them, right? You don't want these numbers depleted by a viral infection like HIV. Still with me? So, you know me, my little stories, little play on words to help me remember these things. So what has always worked for me is that 4H, right? 4, and H for helper, right? Which 4H is also an organization that teaches kids about animals, right? Helps them, right? But I always remember 4H, right? 4 for CD4, H for helper T cell. And then you just divide the 4 in half and you get the 2 for the MHC2. Right? So MHC2 and CD4. That's the match. Right? Just like we learn to match DNA, right? A's with T's, G's with C's. In this case, MHC class 2, CD4, helper T cells. That's the match. That's the communication. Still with me? All right. So moving on, right? So they communicate with each other, right? They start releasing these cytokines, interleukins that talk between white blood cells. So they have interleukin 1 and interleukin 2. And then after this presentation, and one of the things that also can happen, especially for macrophages, remember, they can become super killers when they become activated. This is activation, right? When they communicate with a helper T cell that recognizes that antigen, they're becoming activated. So they may be spurred to make more lysosomes that contain different digestive enzymes that may help them eliminate the pathogen that they're dealing with. But they're also kind of calling in reinforcements right now, right? They're talking to the helper T cell. The helper T cell's like, yep, I know this bad guy. I'm going to go tell everybody else about it, right? Yeah, they're not joking around. Right. And that's pretty much the job of a helper cell, right? He's helping out. He's going to help activate that macrophage, and he's going to help other cells take care of this problem, right? And this is where we kind of split then, right? So in the case of helper T cells, they pretty much help out the B cells. So I'm going to skip ahead in the animation to that interaction. So I'm going to skip over the cytotoxic. I'll come back to it. But here's the B cell. So what do we see on the surface of this B cell? Yeah, we see antigen receptors, and we call these for B cells, what are these? That's an antibody, right? And it's, look at it, it's directly interacting with the antigen. What class of antibodies on the surface of the B cell? IG. Well, the first one you produce. M. When it's secreted, remember, when this B cell gets activated, it will secrete it in that big pentamer mega form. But on the surface of the B cell, it's in the monomer form, embedded in the membrane. No. It's always IgM on the surface of the B cell. It's the first class of antibody we make. It's going to make sense when we look at the DNA. But IgM's first. And therefore, it's the one on your surface of your cell. So what do you think this is, though? This is some other receptor, though, right? This is MHC class 2. Because who is this B cell going to talk to? The helper T cell, the CD4 helper T cell. And that's what they show next. But, do you guys notice? Oops. Oh yeah, the T cell receptor is somehow interacting with the antibody. No. It's supposed to be binding over here, right? And the antigen is supposed to be presented also right here by the B cell. So somebody screwed up in the animation, right? It's not talking to the right things. So, but again, this is kind of a, you know, secret handshake, confirmation, right? This T cell, this helper T cell is telling that B cell, yep, I recognize that guy. He's bad. Yep, you better do what you need to do. And what is a B cell going to do? it's going to now, instead of having it on its surface, right, because then they're stuck there, it's going to actually secrete them. And when a B cell turns into an antibody secreting cell, it's referred to as a plasma cell. And one of the ways to help you remember this, too, oh, they don't show it in this one, but look at that. And so it's this same B cell that proliferates. When it becomes activated, it makes copies of itself. And it makes copies and makes copies and makes copies. So you have thousands, right, of these B cells, and they're pumping out millions, like millions per second, of these antibodies. So in order to do that, what are antibodies? What type of molecule are antibodies? They're protein, right? They're 98% protein. They have a little bit of sugar associated with them, but pretty much they're a protein. What makes proteins in our cells? Starts with an R. Ribosomes. Ribosomes make proteins. But the ribosomes that make proteins we secrete are attached to the rough ER. So the rough endoplasmic reticulum in these B cells gets huge, right? They actually increase their rough endoplasmic reticulum. And so their cytoplasm becomes huge. Hence why they're called plasma cells, right? Plasma, cytoplasm, big. So they just become these huge antibody factories, and they secrete it. Now, depending where that B cell is, right, if it's associated with our mucus, right, it's going to pump out probably IgA, right? If it's in the lymph nodes that we'll talk about later, it's going to pump out IgG. If it's in an area for an allergic individual like me, what is it going to make? It's going to make IgE. Right. The very first one it's going to make most of the time is M, but that's going to switch to those other classes depending on the signals that that B cell gets and its location in the body.
So is that why the IgE and the basophils are kind of linked together because of the lesser information discovered about it?
Yeah, I don't know.
It just made correlation because it says that there's not a lot of information that relates to the IgE and basophils are not as commonly found in the bloodstream.
Right, because that interaction usually happens in the tissue. Thank goodness, right? But it can happen. So he's asking about that relationship between IgE and basophils, and we're going to talk about that in disorders. But yeah, you don't have a lot of basophils in your bloodstream, thank goodness. Because if they're coated in IgE and then you get something in your bloodstream that activates those antibody-coated basophils, then you have the release of histamine, which is a vasodilator. It's a strong inflammatory substance because of the vasodilation that, you know, if you have that happening in your bloodstream, that's when you go into shock, right? That's when we talk about things like anaphylactic shock, when you have that type of reaction happening in your bloodstream. A lot of times it happens in the tissues. Like, so for individuals like me, you know, it happens in my mucous membranes of my nose, you know, or in my skin. Anything that gets into my skin. It's just disgusting. So, makes sense? Good thinking. No. Good thinking. Okay, so these plasma cells, and as they said, right, once a B cell becomes activated, it's gonna, some of them are going to turn into plasma cells and secrete antibodies. What are the rest of them going to do? They become memory cells. They hang out in your tissues, in your lymph nodes for decades, right, remembering the bad guy. So when that bad guy comes on again, right, that activation process and the number of B cells that produce plasma cells happens that much faster. And each time you activate these guys, you get memory cells. What they don't show you here, too, is guess what? When a T cell gets activated, it, too, produces memory cells. right? So whenever B cells and T cells, whenever those lymphocytes, those specific antigen recognizing lymphocytes get activated, you get memory cell production as well. They're like, oh, I want to remember this one. I'm going to keep a guard that knows exactly what this bad guy looks like.
So they only live for like a couple days, right?
Yeah, they believe. And then, so that's why, especially like with tetanus, you want to get boosted. And the reason with tetanus why you want to get boosted is actually your antibodies will actually circulate for like 10 years. It starts waning around 5 for, they found for tetanus. They want your antibody levels to stay high. So you're reboostering those memory B cells, too, in order to get it. But I mean, yeah, I mean, each person varies, you know, so it's an average of the studies that they've done, you know, as to what's effective. But I mean, we don't know that exact answer, you know, and it's, and you really, person to person, it's going to vary too. So there's never going to be an exact answer that like, oh, a B cell will stay around against this infection for 50 years. Might. It just depends too on exposure. And that's why boostering certain vaccines, especially tetanus, are important to make sure, right, that we keep them, right? We don't lose them. Good question. All right, so now I'm going to rewind back to our cytotoxic T cells. So antibodies, as we said, are great, right? Let me finish on them. So they can bind to some antigen, right, and tag it for destruction by a phagocytic cell. What antigen antibody outcome, what's the name of this when it tags it for destruction? Opsonization. Opsonization. All right, so now I'm going to rewind to cytotoxic T cells. So antibodies are great protection, but again, they're just going to recognize stuff outside of our cells or stuff on the surface of our cells. Antibodies do not go inside of our cells. So what if a bacteria or a virus, or what if our cells just starts to behave badly and not do what it's supposed to do? How are we going to detect that? We need to have a cellular level of detection, right? So this is our cellular component of our adaptive. And the major player in that are what? What are these guys? They're a T cell. They're cytotoxic T cells, right? So do they have a receptor? Yeah. Do they have a specific handshake? You betcha. All our normal cells present antigen. MHC class 1. All your cells, including those macrophages, by the way, and dendritic cells, present using MHC class 1. Just dendritic and macrophages and B cells are special in that they also do MHC class 2 because they actually take stuff inside, right? So that they can communicate that to the system saying, hey, I took this in, right? This is an external antigen. Respond to it with an antibody response because this is stuff outside of our cells. Or in the case of a virus, right? Do we want an antibody response? Yeah, because there is a time that that virus is outside of our cells, right? We want to be able to neutralize it, right, before it gets in with antibodies. So usually for a viral infection, you have both a humoral response, but you also probably want this response to what we refer to as cellular. Because when an infected cell is infected by, say, that virus or an internal bacteria, that cell is going to present on its surface. If a cytotoxic T cell comes along and recognizes that antigen, guess what it does to that cell that's infected or behaving badly? It's a cytotoxic T cell. So what does that tell you? He's a killer, right? Sometimes they're referred to as killer T cells. I don't like using that term because then sometimes we get confused with natural killer cells, which are different, right? These are cytotoxic T cells. Or, again, a lot of times we just refer to them by their CD marker. And what's their CD marker? Theirs is CD8. Right? They are CD8. CD8 allows them to communicate with MHC class 1. And how I remember this one is the number 8 almost sounds like to eat, right? And these are toxic cells. They basically eat other cells. And they can eat just about anything, right? So it's the number 1, MHC, number 1, right? Very important to be able to recognize when our own cells are infected and that we destroy them. We do this by our cytotoxic CD8 T cells. So again, once a cytotoxic T cell is activated, it'll proliferate. So we've got a whole bunch of these guys that are running around looking for our cells that may be infected or behaving badly. And they're going to kill them. And we're going to produce memory cells, right, of these cytotoxic T cells. So if this happens again, they'll respond that much quicker. Same thing with helper T cells. Once they become activated and they proliferate, so you get a whole bunch of them that can go talk to B cells, you got memory ones of those guys too. So all the major players, right, all the T cells, all the B cells, if they become activated, we're going to produce memory cells. So we can do this all over again, but much faster if it happens again. So that first exposure we refer to as our primary response. The immune system remembers, right, by producing those memory cells. If accounted again, right, our second or third exposure is much stronger because of that memory component. 2 basic strategies, right, especially for bacteria that mostly are extracellular, right, don't go inside. We're going to produce antibodies, right? Remember, the humoral immunity is antibody immunity. We're going to make antibodies against bacteria. We're going to make it against toxins. We're going to make it even against viruses, right? Because there is a time that they're outside of our cells, and we want to stop them from getting in. All right. So they're saying in the bloodstream, basically saying viruses that haven't gotten into our cells yet. Antibodies are going to help protect us against. Right? These antibodies stay in our tissue fluids, right? They'll circulate through our blood. They'll get secreted into our secretions. Even under inflammation situations, those antibodies will go into that area. B cells will actually even go into that area, turn into plasma cells, and produce antibodies on site. Right? Those B cells will be there, right in the tissue, producing antibodies until the bad guy is gone. Cell-mediated immunity deals with stuff that gets inside of our cells. So very important for viral infection. But also there are some bacteria that get inside that are intercellular pathogens. So having a cellular response is important. And then again, too, because we know the difference between healthy self and non-self, damaged self, this will also take care of our own cells that may be cancerous, right, behaving badly. Both of these systems can be activated by vaccination. When you get vaccinated, yeah, a lot of times we're going to see if the vaccine was effective by checking your antibody titer, right? How many antibodies did you produce? How many are in your blood? But what we really can't test, that I'm aware of anyways at this point, is if you've gotten a good cellular response. But they've also, are engineering and trying to make certain vaccines so that you do get a good cellular response as well. And that's why they actually give you a whole virus that will replicate, but doesn't usually have all the components to make you sick. Right? So that's why the chickenpox vaccine, y'all, is chickenpox virus, pretty much. Right? It's It's just they're giving it to you in a shot. And again, you're going to make antibodies and you're going to do a T-cell response. When we can give you, especially a pathogen that's going to replicate inside of your body, so if we actually give you a bacteria, a whole bacteria, that's going to actually grow, but isn't as lethal as the real thing, then you're more apt to produce a much better immune response to it, right? Because the initial dosage we give you is small, but it itself gets copied in your body. And so that helps with making sure you make a good response to it. So this is one of my favorite diagrams. I think they do a really good job of presenting the major players in cellular and antibody or humoral immunity. So at the very forefront are your antigen-presenting cells, right? So say a dendritic cell. He's got to present that information to T cells. Because a lot of times, although B cells can do this on their own, they don't do it as effectively, and they don't switch from IgM. IgM is the first one they produce. For a B cell to produce any of those other antibody classes, it needs a helper T cell to help it make that switch. I really wish this congestion would just go away. I'm about done with it. Done with it. Ugh. I'm still here though, so the immune system's working, right? Or not. Could be at this point. It could be allergy now. So that's the unfortunate thing for me. So, but for T cells, it's got to be a cell-to-cell interaction, right? So an antigen-presenting cell must present this and activate these T cells. Now, if it's a helper T cell that is being activated, it's because that antigen-presenting cell is presenting that antigen in which MHC. If we're talking to a helper T cell, too. Right, we're doing antigen presentation. If we're talking to a cytotoxic T cell, it's presenting with MHC class 1. Right? And that's where we get the cytotoxic T cells that will interact with any cell that's got MHC class 1 on it and is presenting that same antigen. Right. And again, whenever lymphocytes, B cells or T cells are activated, they're always going to proliferate. Right? You don't want just one guy that recognizes the bad guys. You want as many as possible. Right? So activation, proliferation, then go do your job. Right? And in that proliferation, there are going to be effector cells, the ones that go out and do the job. And then you're also going to produce what? Not shown here. Right? It's activated. We're proliferating. Are all of them effector cells? All of them are going to go out and do a job? No, you got memory cells. Right? You got memory cells. So remember, T cells, helper T cells, can also activate macrophages. Right? But they are definitely necessary for activating B cells, especially if we want to switch to a different antibody class other than IgM. And of course, this isn't IgM, right? Because when IgM is secreted, what does it look like? It's that big pentamer, right? That huge complex. So we already started talking about this, right? Each one of these cells has their own special, unique receptor. For the B cell, it's called its B cell receptor, but in fact, it's IgM class antibody. It can interact with antigen all by itself, right? T cells, on the other hand, have their T cell receptor, right? And each one's unique to that T cell as to which antigen it can recognize. Each T cell also has that CD marker, and that determines whether it's a helper T cell or a cytotoxic T cell, and therefore what cells can it communicate with. And again, it's cellular though, right? T cells only talk to other cells, only communicate with MHC, whether it be 1 or 2, depending on which T cell you're talking about. They cannot recognize antigen all by itself. It has to be presented to them by our cells. So how and where does all this happen, right? So we actually have a vascular system that is specifically for draining away the bad guys. And we refer to that as our lymphatic system. So unlike our circulatory system, it does not circulate, right? You can almost think of it as the drainage system, okay? The sewer system, right, for our body. Because you don't want the bad guys getting into the bloodstream and getting a free ride through the whole body. So when we have inflammation, right, fluid goes into the tissues. Well, eventually we need to drain that fluid back out of the tissues. I just see Hammond right now, right? I see all the water draining out of northern Louisiana, right? And as that drains, well, all that water, right, or all that fluid in our tissue, is it clean? No, right? So you just watch the news right now. That water is nasty, okay? Would you want all that water that was in our tissues to just go right back into the bloodstream? No, because what could it have in it? If you got a cut, right, and bacteria got in, and say we don't eliminate it, that fluid goes right into the blood, boop, just got a free ride to a whole bunch of cool places to go eat you throughout the body. Not a good idea. So instead, that fluid that goes into your tissues drains into your lymphatic vessels. It's a completely different vascular system. It starts in your tissues, and it drains, and as it drains, it filters it all throughout your body until all those vessels come together into 2 ducts, your right and left lymphatic ducts, that dump then into what? Your veins, your subclavian veins, right? But before it got there, right, it was drained by lymph nodes. Guess what's in the lymph nodes that's going to gobble up any bad guys? That macrophages, dendritic cells, right? And then also B cells are there and T cells are there, so they can become activated if something comes on board. And so your lymph nodes then too will secrete antibodies that go into the lymph, that then go into your blood and go and circulate throughout your entire body. Now your primary lymph organs are where your lymphocytes, where your white blood cells are born. And where is that? In your bone marrow, right? All your blood cells come from the bone marrow. So that's a primary lymphoid organ. It's the beginning of it, right? The other one for your lymphatic cells, your lymphocytes, is for your T cells. Because your T cells, remember, they have to know, they have to communicate with our cells. They have to know, is this my cell? Is this my buddy, right? Or is this a bad guy? Or is this one of my cells behaving badly? We have to have that level of communication. So they need an extra level of education. Anyone know where that happens? Happens in the thymus. So that's why T cells are called T cells. They're named after where they get their secondary education, you could say. All right, one last one and then I'll let you guys go. The spleen. What does it actually filter and capture pathogens from? Your blood. And that's why this is the only organ, if you lose, you don't have to get replaced. Because that's its main function. And although it's very helpful for helping us stay alive, it's not necessary to stay alive, right? Your liver will take over the function of helping recycle your red blood cells, which is one of the main things that your spleen does, other than getting any pathogens that may have gotten into the bloodstream. So they're chock full of macrophages and dendritic cells and B cells and T cells as well. Your spleen is. So your spleen, right, all the places where your lymphocytes live, all those lymph nodes, right, those are your secondary lymphoid organs. Your tonsils, your adenoids, your appendix. And that's because they're organized to the organ level. Does that mean you don't have B cells or T cells anywhere else in your body? No, right? They're in a lot of your tissues, especially associated with your mucous membranes and your skin, right? Because those are major areas where bad guys could get in. It's just not organized to the organ level, right? These are the only organs. All right, so we'll pick up here Tuesday, and I will let you guys know, hopefully by the end of the day today, when the test will be, because I got to reschedule.
The thing on Monday, when you does it have a time?
Yeah, it's at 2:30.
2:30? Thank you.
It does have the time. It's right there.